The speaker is Dr. Chen from the Genome Institute, Singapore. He is now a senior research scientist at this institute, and today's talk is a highly specific multiplexed RNA image in a tissue with a split fish. Dr. Chen, please.
Thank you for the introduction, and thank you for giving me the opportunity to talk about this work in this seminar. My name is Kok Hao, and I'm going to tell you this story about improving this RNA imaging. It's a bit of a switch gear from the sequencing, but hopefully we're all looking at the same thing. So just brief introduction, right? That we are all interested to identify the tissue and cellular identities in different systems. And in the single cell sequencing, you can get really very detailed molecular information. And what we are interested in is to find out where these cell types are located in the tissues. And there are a host of technology, and one of them you just heard, the GeoSeq, to try to get to spatially resolved transcriptomics. And not just to see where the cells are located in the tissues, the different cells, how are they organized, but also hopefully, with enough resolution, you can look at the subcellular distribution, whether they are located at the nucleus, the edge of the cells, and if they are polarity associated. And then, of course, there are a host of technology to sequence the RNA. You can use in situ RNA capture like CytSeq or 10x ST, or you can even try to sequence the RNA inside the tissue. And the focus of this talk is on using a multiplexed which is called like a SIGFISH, MERFISH, and it's the core technology is using the single molecule fish, which is to label the RNA species with a lot of probes so that they show up as diffraction limited spots in your image. And then the key idea to do multiplex fish is to use the serial hybridization based barcoding. So in addition to labeling the probes, labeling the RNA with the probes, we also label them different groups of RNA with readout probes so that they are fluoresce in different types. And by doing this hybridization and washing cycle, and then we align the images, and then based on the sequence that they are on and off in different images, we can tell which RNA is which from the image. And from this, we saw that the counting is actually pretty accurate if we compare it to RNA-seq. And the nice thing about this is that it scales pretty favorably with the number of hybridizations. In fact, very recently, from the Kai lab and Xiaowei lab, they showed that this technique can be extended to genome scale. So now it's almost comparable to doing sequencing. And there are many applications that we can envision to map the different cell types, image chromatin, or construct spatial cell atlas. So looking back when we were trying this technique, I thought we were quite crazy because we were actually labeling the tissues with a lot of highly complex pool of sequences, right? We're dumping 100,000 different sequences, and what were we thinking if we can see anything at all? And here showing is an image, the typical image on an uncleared tissue. You can see that the RNA are kind of concentrated in the cell bodies, and this is just a raw image showing the whole camera intensity and process. And we always wondered, how does it look like? What are all these other stuff? Are they RNA, autofluorescence, non-specific binding? And it's also very difficult to optimize for so many sequences, right? We can try to maybe make the length longer so that the melting temperature is narrower range, and but it's hard to optimize. And if the length is too long, then it will be non-specific, right? Because the thermodynamic gain of many base pairing will override the penalty from a field mismatch. So one of the very nice solution is to do gel embedding and tissue clearing. So as shown here, not only you can eliminate the autofluorescence, but you can also eliminate non-specific binding to proteins and lipids. And with this, people have been able to scale to genome. But this technique also has some shortcomings, like it cannot remove non-specific bindings to other RNA, and the clearing chemistry might not work for all the tissue types that people are interested in, and it also takes some time to clear the tissue. So we are wondering if there are a simpler alternative. And one thing that we thought of is to use a coincidence detection via complementation hybridization. So a very simple example is a split GFP. So in the cell, you can have the GFP fragment at very high concentration, but they don't contribute to background because they only fluoresce if the two fragments are on target, so that now you get very good signal to noise ratio, right? So we're wondering if this same concept can be applied to oligo probes, and where we have, now instead of one probe, we have two probes, and we need both probes to be on target before the fluorescently labeled bridge probe can bind and give out fluorescence. And this is actually very nice because now with a two-step hybridization, we decoupled the affinity and the specificity. When we look at the literature, actually this idea of using two probes is there already. We can look in the RNA scope where they have this two Z probes here, or HCR 3.0, the more recently one, look like this. And then there's the snail probes where they use in the in situ sequencing. Because there's so many different designs and the protocol is also complex, right? In addition to this coincidence detection, there's also amplification. So we are wondering which one of them are optimized for specificity, and can we apply these to multiplex fish? So we try to optimize this ourselves. First thing we were thinking that if we want the probe to be specific, right, then the unpaired probe has to produce no signal. If one probe by itself, it has to produce no signal. So we figured that this length here, the bridge length, has to be short, right? So we titrated it and we found that maybe 9 nucleotide is good. It doesn't produce a signal by itself. And then we went look into the different configuration. We look in the different permutation way to pair them up. And to our surprise, actually, one of the configuration works really well. This configuration seems to have, no one is using this configuration. So this is just a simple circle configuration. And the bridge probe can bind with very high efficiency. It's almost as bright as just using the conventional probe sequence. And then just to finish the optimization, we also try to shorten the bridge sequence, and we found that if the sequence is really too short, then it also impacts the efficiency. So we found that this region here is very ideal. And with this configuration, which we call splitFISH, first thing we try to figure out is, does it show improved specificity? So this is a co-localization experiment. It's very common. So what we did is with one color Cy3, we use a lot of probe and target this mRNA in the Cy3 channel. And then in the Cy5 channel, we varies the configuration. And we found that indeed with the circular configuration, it has the best specificity indicated by high degree of co-localization between the two channels. And perhaps this is not surprising, right? Because it's giving the brightest signal. But not only we want our probe to be on target, we also want the probe to not give signal when it's off target. So we went into the literature and we looked, and there's actually a very nice example here. So in this study, the authors were trying to find out the localization of non-coding RNA, and to be robust, they actually did this two-color co-localization test on their non-coding RNA. And one of the RNA, X-locked010514, failed their co-localization test. And they were wondering why. And when they went in to titrate each oligo, they found that, actually one of the oligo is giving this false signal in the nucleus. And when they remove only one out of the 50 oligos that they use, the true localization of this non-coding RNA is revealed. So we tried to see if we can use splitFISH to eliminate this off-target signal. So first, we repeated their result where we see that this sequence is indeed giving the false nuclear localization. And then we try to pair all the sequences with adjacent sequence and design splitFISH probes for them. And we found that indeed, with the splitFISH, we can eliminate the false localization signal. Just a quick recap, We found that the circular configuration improves specificity without sacrificing efficiency. And we are really trying to see if we can multiplex this splitFISH. So we did some optimization, including trying to adapt it to oligo pool so that we can stain a lot of target sequence. And then we optimize the chemistry so that we can do sequential hybridization and barcoding. So when we when we tried to do this in the cell, in the tissue, actually, we were really surprised. When we first saw the image, I thought we mixed up the sample. Now, are we staining single molecule fish? But this is indeed the image when we stain 317 genes, where we use a pool of like 50,000 sequences. And the RNA is just crystal clear in the samples. And we see that they almost look like we have cleared the samples using chemical techniques. So we went on to optimize the hybridization condition. We showed that we could indeed stain the cells and remove the signal repeatedly and very robustly. And because we have this technique, right, and maybe we thought, well, we could apply this oligo pool to different organs. And since we might not need to clear the tissue, we can readily adapt it to different tissue types. And this is indeed what we see when we apply to different tissue. We see these beautiful images. For example, in the brain, we see different layers, and same with kidney, we see this zonation patterns indicating the cell types. And also in the liver, for example, the different cell types occupying different portal veins versus the central veins, and also different follicles maturation stages in the ovary. And we think that the RNA counting is also accurate because we try to compare it to bulk RNA sequencing. And the count is all correlated well to the different tissue types. And finally, we try to compare it to conventional multiplex fish in uncleared tissues. And we see that using splitFISH indeed reduce the blank counts or the misidentification rate. And with that, we are very excited to apply this to more tissue types and to scale up to more genes, or combine it with signal amplification or tissue clearing to get even more accurate measurements. But as we are doing this, found that actually there's a limitation to this splitFISH technique. If you have some idea about this, I'd love to discuss with you. And the limitation is that because we are designing the bridge sequence to be very short, right, for the coincidence detection to work, we actually have very limited multiplexing capacity because now we only have space to put like one barcode right here versus, the conventional design where people scale to the genome by using 4 barcodes. And with that, I just want to acknowledge all the people who have helped in many ways, especially Jolene and Nigel, who did a lot of this work. And thank you all for listening, and I'll be happy to answer questions that you may have.
Okay, thank you, Dr. Chen. It's very nice talk. A new method is very nice. So we have questions. So this is now is still the questions for Aibin. Okay, if no new question coming up, I will ask a good question. So with your fish, you said you detected about 317 genes. So this is a hybridization each one of the probe, or you hybridize with one pole of hybridization probe? You do the series hybridization or the can you not use a full screen?
Stop sharing. Yeah.
So my question is, when you did all this more than 300 probes hybridization, so the probe is applied once together or the series hybridization? You did this.
Yeah. So it's a very good question. So what we have to do is we will apply all the probes at the start, and then we will apply the bridge probe later sequentially. So the bridge probe and the probe with the fluorescent, the readout probes, they will apply it sequentially so that we can control which group of the RNA is labeled. And, and then from the on and off, we try to decide which RNA is which RNA. Okay. Yeah. So it's a 2 or 3-step process now. So we have first a pool that labels everything. And then we have some probes that bridge them, and then we have some probes that read out. Yeah.
Okay. So for each gene, you designed 2 of the probes with first pool, right? And then you can have a bridge with connected with the other, these 2 together, right?
Yeah, to connect them.
Okay. Okay. Okay, this is a quick question related to Dr. Jing's. Is detection, a quick question related to Dr. Jing's, is detection of efficiency more related to the expression level or the sequence complexity?
Very interesting question. So we believe that the detection efficiency actually is directly related to the number of probes that we use to target the RNA. So it should be independent of the expression, the amount of the gene, but there is a limit because when the expression of the gene is very high, then it cannot be resolved under the microscope. So there is a limit. In fact, I think it's good if we can, detect some lowly expressed genes, which could be interesting in certain systems. But because of this, it's also a limitation of the technique because if the gene like very short, like microRNA or something, then it becomes extremely impossible to detect. Because now we have to target with like 1 probe or 2 probes, which will not guaranteed to be specific. Yeah.
So Evan still says, particularly when you design the probes, this is quite difficult, right? So I was heard this technique is quite difficult to mask for the normal lab. They need a very sophisticated microscope. Is this true?
Actually, it's not very sophisticated. The microscope itself, it doesn't have a very high demand. Like you can use a wide field microscope, which is kind of what we use. People have also used confocal microscopes, but it does take a bit of effort to try to keep the system stable and try to exchange the fluid because we now need to do the hybridization many rounds. Yeah. And align the images back. Yeah. We have a question from Joe. He was wondering how can you get the varied split probe oligo pool? Yeah. So that. Right, so you got it. So the split probe, we have to obtain it from the oligo pool because it's a very high diversity. So there are quite a few companies now offering this oligo pool product where they can synthesize like sequences, up to like hundreds or almost 1 million sequences. Yeah, I hope that answered your question.
Good. We still have a few minutes. We can wait 1 more minute to see whether they have a further question for Dr. Chen's talk. It's very nice. My question is how large, because you lastly you showed the ovaries. That is the mouse ovary, adult mouse ovary, or liver, the tissue detection. It's adult. Yeah. Okay, so
it's from wild type adult mouse. Yeah.
Very nice.
I think the area that we managed to image is about like a few millimeters, but it does take more and more time to image a large area, right? We also need the resolution to resolve the molecules, and then we have to scan and scan, so it does take quite a bit of time.
That's true. That's true. Okay, if no further question, I would like to thank all the speakers and also the audience to have our session. Very nice sessions. Okay, thank you. Thank you both of Andy and Dr. Chen. Okay, hope we can really meet sometimes offline, right, to discuss more. Because still online have some communication is not that easy. Okay, good.
Yeah.
Okay, so we end our session. Okay, good.
Yeah. Thanks. Thank you.
Thank you.
Thank you.